In this presentation, I will be talking about photoperiodism, a topic from the paper Plant Physiology 2. In this module, we shall study how light is perceived by plants, how the duration of light is measured, and how the signal is communicated to bring about the induction of flowering. Plants require light for growth and development. Through photosynthesis, plants are able to convert light energy into chemical energy, which is used for their survival and growth. However, light also plays an important role in regulating developmental processes, like flowering, for example. Plants can sense and respond to the duration of light in a 24-hour period, in other words, the day length. This can be seen when trees burst into bloom in specific seasons, when the day length is appropriate for triggering the flowering process. In this module, we will first have a look at photoreceptors and study how they transmit the light signal. Then we shall study the role played by these photoreceptors in regulating the time of flowering in plants. Perception of light requires photoreceptors. Plants have three types of photoreceptors that can sense red and blue regions of the visible spectrum. The red light photoreceptors are called phytochromes, and the blue light photoreceptors are called cryptochromes and phototropins. The photoreceptors exist as pigment protein complexes and show the presence of photosensory domains which bind the chromophores. The chromophores are phytochromobilin P psi B in phytochromes and flavin adenine nucleotide or FAD in cryptochromes. The blue light receptor in phototropins is flavin mononucleotide or FMN. Each photoreceptor also has regulatory domains that transmit the light signal to bring about a response. Of these photoreceptors, phytochromes and cryptochromes play a role in regulating photoperiodic responses. Let us understand what happens to the phytochromes when they perceive red light. There are 5 types of phytochrome monomers, which means that there are at least 5 genes coding for the phytochrome proteins. The phytochromes are organized as homodimers, as in the case of phytochrome A, or heterodimers, as seen for phytochrome B, C, and D. When red light is sensed by the phytochromobilin molecule of phytochrome, intramolecular rearrangements in the chromophore take place as seen in the figure. This in turn causes changes in the protein conformation and brings about autophosphorylation of phytochrome. The form of phytochrome in dark conditions, also known as far red conditions, which has an absorption maxima at 730 nanometers, is the PR form, which is converted to the PFR form when red light having an absorption maxima of 660 nanometers is sensed. In the PFR form, nuclear localization signals or NLS present on the protein part of phytochrome are exposed. The NLS are recognized by nuclear import proteins called importans, which chaperone the PFR form to the nucleus, where it brings about regulation of the light response genes. Migration of phytochrome to the nucleus can be visualized by making transgenic plants that express fusion proteins of phytochrome with green fluorescent protein or GFP. Green fluorescence, due to the expression of phytochrome fused to GFP, is observed in the cytoplasm of leaf cells when plants are placed in dark. And in plants exposed to light, the fluorescence is observed in the nucleus. The second type of photoreceptors, namely cryptochromes, are blue light receptors. Cryptochromes are present in plants, animals, and fungi. These proteins show structural similarity to a DNA repair enzyme called photolyase, but cryptochromes lack any DNA repair activity. There are 2 types of cryptochromes present in plants, namely Cry1 and Cry2, both of which are seen to be present in the nucleus. These monomers consist of a photosensory domain called photolyase homology region or PHR towards the amino terminal, to which 2 chromophores flavin adenine dinucleotide or FAD, and methynyl tetrahydrofolate, also called MTHF or pterin, bind. The C-terminal extension or CCT domain, which is also called the DAS domain, plays an important role in transmission of the light signal. Autophosphorylation of cryptochrome protein is required for its activation. Blue light of around 450 nanometer wavelength causes FAD reduction to form FADH2, while MTHF acts as the light harvesting antenna pigment. Exposure to darkness causes oxidation of FADH2, and this makes the cryptochromes inactive. Such reversibility in active and inactive forms is seen in Cry1 but not in Cry2. As seen in the figure, the two monomeric cryptochromes interact at their PHR domains to form the functional homodimeric forms. The CCT domain carries a nuclear localization signal and a kinase domain that brings about autophosphorylation of the cryptochromes on activation by blue light. Several accessory proteins are also known to bind to activated cryptochromes to bring about blue light specific responses, as we shall see later. Besides photoreceptors that sense light, the duration or time of the light period is measured by a mechanism called the circadian clock. The clock consists of input pathways, central oscillators, and the output pathways. Identification of these proteins and studies on their roles was made possible by studying mutants that show abnormal light responses. Further, overexpression of these proteins in transgenic plants was seen to lead to a loss of the oscillatory behavior of these proteins and hence a loss of circadian rhythms. We shall see how these pathways operate. Two proteins have been identified which show an oscillatory behavior in expression during a 24-hour period. These proteins belong to a class of transcription factors called MIBs. They are the late elongated hypocotyle or LHY protein and the circadian clock associated 1 or CCA protein. These proteins are induced by phytochromes and cryptochromes during the day. The clock proteins in turn induce two other regulatory proteins called pseudo-response regulators, PRR9 and PRR7, that negatively regulate the expression of CCA1 and LHY. Hence, LHY, CCA, and PRR9, PRR7 form a feedback loop. In the dark, when phytochromes are in PR form and do not activate LHY and CCA1, the PRRs will close this loop. So the duration of expression of these oscillatory proteins is modified by phytochrome and cryptochrome to synchronize the internal clocks to the period of light availability. In the evening, the clock proteins LHY and CCA1 are no longer active. This allows expression of two other oscillatory proteins, gigantia or GI, and the timing of CAB expression TOC1. Expression of gigantia peaks in the late evening and it forms a complex with another protein, Zeit loop or ZTL, which is an FPOX protein and brings about proteolytic degradation of TOC1 in light. In the dark, gigantia or Zeit loop are inactive, which allows TOC1 expression in the dark. TOC1, like CCA1 and LHY, is an oscillatory protein. Other regulatory components of the clock genes and the output pathways have been subsequently identified, which show that there are several overlapping loops operating during the morning and evening besides these two loops. Hence, phytochromes and cryptochromes entrain or synchronize the clock to light duration by regulating the expression of clock proteins. Depending on the coincidence of clock and light inputs, an output signal is generated that leads to induction of flowering. Having understood the mechanism by which phytochromes and cryptochromes perceive light and measure its duration, let us now see the nature of physiological and developmental responses that are mediated by phytochromes and cryptochromes. Phytochromes are involved in regulating seed germination, seedling growth or photomorphogenesis, shade avoidance by which plants alter growth to get sunlight, and flowering. Cryptochromes regulate photomorphogenesis and flowering induction like phytochromes, but also have some exclusive effects like regulation of stomatal opening and hypocotyle bending. Hence, some responses of phytochromes and cryptochromes overlap, while others are distinct. Of these developmental responses, the flowering response is an important photoperiodic response involving both the photoreceptors. We will now see the effect of photoperiod on induction of flowering. Plants show variation in their day length requirements for transition to flowering. This was observed way back in 1920s by Dr. Garner and Dr. Allard of USDA laboratories in Beltsville, USA. They put forth the theory that day length was a determining factor in flowering induction. Flowering species fall into two photoperiodic response categories, namely the short day plants and the long day plants. Short day plants require day length that is lesser than a critical number of hours for induction of flowering or for acceleration of flowering. More importantly, these plants require a long, uninterrupted dark period. If the dark period is interrupted by a flash of light, it prevents flowering in the short day plants. The critical period of light or day may vary from one plant species to another. Long day plants, on the other hand, require day length that is greater than a critical number of hours for flowering induction or for acceleration of flowering. If a longer dark period is interrupted by a brief light period, the long day plants will also show flowering. Some plants flower irrespective of photoperiod and are called day neutral plants. However, it must be realized that there are other factors besides light that affect transition to flowering. These include temperature, age, hormones, and sugar signaling. Let us now study the photoperiodic response of flowering in the long day plant Arabidopsis. Arabidopsis plants show flowering when the day length exceeds a threshold of about 16 hours. Phytochromes play a role in regulating flowering when the photoperiod is optimum. During day, the active form of phytochrome interacts with a clock protein like CCA1 or LHY that shows oscillatory expression as seen earlier and synchronizes the clock with light availability. This interaction between phytochrome and the clock protein brings about an increase in transcription of a flowering signal gene called constans or CO. CO is a transcription activator which transmits the signal via another regulator of flowering called flowering locus T or FT to the flowering meristems. Besides acting at the transcription level, phytochromes also affect the stability of CO protein. The active PFR form of phytochrome is known to bring about proteolytic breakdown of CO. However, the PFR form of phytochrome A is short-lived and does not show photoreversibility. Hence, in the evening, the PFR form of phytochrome A does not degrade CO since the PFR form is unstable. On the other hand, the PFR form of phytochrome B degrades CO protein if it is expressed in daylight. Hence, phytochrome A and phytochrome B appear to show opposite effects on the stability of constans protein. However, these phytochromes act at different times of the day and hence CO protein accumulates and is stable in the late evening. Cryptochromes show a similar effect as phytochrome A in preventing proteolytic degradation of CO. Other proteins called suppressors of phytochrome A or SPA proteins play a role in degradation of CO protein during the dark period, as we shall see later. After phytochrome and cryptochrome, regulate the expression or stability of CO in the leaves, which are the organs of photoperiod perception. CO activates the expression of a transcription factor called flowering locus T or FT, which is a long-distance signal for transition to flowering. FT moves to the shoot apical meristem through the vascular system and, along with another transcription factor FD, transforms the shoot apical meristem to an inflorescence meristem by activating transcription factors like apatala-1, which plays a role in formation of floral meristems. Under short day conditions, another protein which is related to FT called centroradialis or ATC is induced and is a flowering inhibitor. Like FT, ATC also moves from the leaves to the shoot apical meristem, and along with FD, it represses the expression of apatala-1. This leads to inhibition of flowering under short days in Arabidopsis, which is a long day plant. Now let us see how cryptochromes regulate flowering. Cryptochromes mediate flowering in a similar manner as phytochrome A and opposite to that shown by phytochrome B. In the dark, an E3 ubiquitin ligase called constitutive photomorphogenesis 1 or COP1 is activated by the SPA proteins. Activated COP1 ubiquitinates CO protein, which brings about the proteolysis of CO. This leads to an inhibition of flowering induction. In light, the active form of cryptochrome binds to SPA and prevents SPA-mediated activation of COP1. This leads to an induction of flowering. Besides acting at protein level, cryptochromes also activate transcription factors called cryptochrome interacting proteins or CIBs, which bring about the direct induction of FT. The process of flowering time regulation in a short day plant differs from the process taking place in the long day plant like Arabidopsis. Let us now see how flowering is regulated in the short day plant rice, which flowers when day length is short and the length of the dark period is long, about 14 to 16 hours. Rice also uses the same photoreceptors and clock components for regulating flowering. Under long days, activated phytochrome B in the PFR form inhibits the CO ortholog in rice called heading date 1 or HD1. Inhibition of HD1 prevents expression of the FT ortholog in rice called HD3A. Hence, flowering is not induced under long days. In short days, HD1 expression is no longer under phytochrome B control since it peaks in the dark. Hence, HD1 can now induce the expression of HD3A, which triggers flowering under short days. Cryptochromes are also known to induce HD3A expression through another regulatory protein called early heading 1 or EHD1. However, the mechanism is not well understood. Flowering in day-neutral plants like tomato also use the same components that induce flowering in Arabidopsis or rice, but the regulation of these components is not light-dependent. An ortholog of FT called single flower truss or SFT provides the signals for flowering induction. However, SFT activation is independent of photoperiod. Hence, light signals interact differently to regulate the flowering response in short day, long day, or day neutral plants. However, the downstream regulatory factors that lead to the formation of floral meristems are similar. Action of these downstream genes is independent of the photoperiod. We shall now summarize what we have learnt in this module. We saw that photoperiodic responses of plants relate to duration of light. A striking light duration dependent response is flowering. Secondly, we studied two important photoreceptors, phytochromes and cryptochromes, that sense light. The photoreceptors, along with the circadian clock system, measure the duration of light or the photoperiod. Finally, we looked at the components which signal the flowering response, namely proteins like constans and flowering locus T. These components are similar in long day, short day, and day neutral plants, but the mechanism by which the expression of these components is regulated differs. So, when you see a tree burst into bloom in the spring season, or rice flowering when day length reduces in September, October, or wheat or mustard flowering during March and April when the days are longer, remember that there are a lot of intricate regulatory mechanisms that make this possible. Thank you.